Good morning. First of all, I would like to thank the invitation of the GARD Symposium. In this opportunity, we'll talk about the role of vaccines in pandemics and outbreaks. We will talk about epidemics history, vaccines and disease, what is a vaccine, how do vaccines help us, and developing vaccines in our century. The crucial role of a vaccine play in protecting our health that come into a sharp focus. During the pandemic, hope comes in the form of a vaccine, and scientists are working to develop a vaccine that will protect us against an infection. The vaccine has an active ingredient. It produces immunity that is a harmless form of a bacteria or virus which cannot cause disease. The role is to introduce antigens to your immune system. The active ingredient is either the whole bacteria or virus, which has been inactivated for attenuated or broken down parts. Now, also scientists use genetic material like DNA or RNA. Water is the most abundant ingredient in a vaccine. Vaccines have a very small amount of a substance added to them to help create a stronger immune response. These substances are called adjuvants. They are particularly useful for people that have lower immune responses. For example, squalene, oil, aluminum hydroxide are used as an adjuvant in these vaccines. Very small amounts of preservatives are used in vaccines to maintain the quality and to ensure the vaccine is safe to be transported and stored. These preservatives are added to vaccines to prevent unwanted contamination. Also, stabilizers stop the component during transportation and storage. The history of immunization is not only full of heroes, but also full of villains. And our successes are tempered by tragedies. The Greek historian Thucydides, who survived catching the plague of Athens during the Peloponnesian War, described the devastating epidemic of what may have been typhus or bubonic plague, which killed hundreds of thousands of people. He recognized then the first recorded description of the concept of immunity because him and other people survived. Jenner, who works on smallpox, violated a large number of people in this parish. Some developed mild smallpox, some severe, but a small number developed no rash. Jenner noted all those who developed no rash had previously had cowpox. Some were milkmaids or had caught cowpox from milkmaids. That was the origin of the vaccine. In 1789, Jenner injected cowpox into an arm of an 8-year-old boy, James Fitz, which lanced cowpox vesicles of Sarah Nelmes, who was a milkmaid. 6 weeks later, Jenner inoculated James with material taken from a smallpox victim. Thankfully, James survived. Years later, Pasteur developed a vaccine against anthrax. In 1982, he gave 25 sheep anthrax vaccine, his anthrax vaccine. 25 controls had no vaccine. Weeks later, all 25 controls died, and the 25 vaccine sheep survived. In the 20th century was the golden age of immunization. In 1884, Klebs and Loeffler described Corynebacterium diphtheriae. In 1888, Ruch and Yersin showed that filtrate of Corynebacterium caused diphtheria-like disease when injected into laboratory animals. Inactivating this diphtheria toxin led the diphtheria toxoid vaccine. The first human diphtheria toxoid vaccine was developed in 1923, and years later, in 1949, they were combined with a whole cell pertussis vaccine that was made by using kill Bordetella pertussis to produce the DTP that we know today as the triple vaccine. Therefore, the COVID-19 pandemic reminds us that we share the same disease pool with both wild and domesticated animals, and the better we protect them, the better we protect ourselves. Due to the long history of plagues, we understand that we are facing and know that vaccination will be a key part of normal health and living being fully restored. So, the ideal vaccine will produce some immune protection without causing disease, generating a long-lasting immunity, interrupting the spread of an infection, safe and without any side effects, confirmed by the clinical trials. The spread of an infection within a community is described as a chain, which is a very interconnected steps that describe how a pathogen moves about. That's the way how disease rapidly spreads across nations and communities. It may challenge the ability of public health systems to prevent and to control the spread of disease, especially in resource-limited countries. The mechanism of classic vaccines go with immunologic pathways. Inactivated pathogens contained in classic vaccines are phagocytosed by antigens presenting cells and presented to native T cells. Antigens presentation triggers the activation and proliferation and their differentiation into different subsets of the T cells, CD4, CD8, and a group of memory of T cells which will persist in the circulation and the tissue for a long time. Will confer immunologic memory after the exposure to the same pathogen. For their part, these cells get activated after direct recognition of the pathogen and contact with a CD4. That's why that was previously activated. The specific B cell will undergo clonal expansion and give rise to plasma cells which produce specific antibodies. Nowadays, scientists add of epigenetic and or metabolic modulators to vaccines as a new approach to increase efficacy. While the combination of antigens and adjuvants has been proven useful developed, the current generation of vaccines, the pharmacologic modulation of the metabolic, the epigenetic mechanism involved into adaptive and the innate immune cells after vaccination can potentiate, fine-tune, and improve responses to vaccine. Such modulation can also use the potential mechanisms in the design and development of a new vaccines and vaccination strategies. Vaccines need targets and platforms. For example, the vaccines against SARS-CoV-2 needs specific gene fragments called RF, RNA-dependent RNA polymerase, PL-Pro, papain-like protease, etc. Those are important to add a plus to the vaccine. Scientists then conduct laboratory research to test their idea for a vaccine candidate. Sometimes this testing occurs in animals. This is considered the research and discovery stage. Before a vaccine can be tested in people, a company of researchers perform additional laboratory research and tests in animals in order to obtain information about how the vaccine works and whether it is likely to be safe and works correctly in humans. These tests are known as the preclinical phase. After start the manufacturing and development and the phase 1 research, a phase 1-2 research, and a 3 will occur. Finally, the regulatory review of starts the vaccination in people. In conclusion, the vaccination plays an important role in a pandemic planning and response. The possibility of develop an effective vaccine for a novel pandemic virus is not assured. However, as we have been seeing in SARS-CoV-2 vaccine development, with sufficient resources and global focus can be achieved in a relatively short period of time. Sadly, even then vaccines are available, there will be scarce. Thank you for your attention.